Hi guys, welcome back. In this video we are covering starch, cellulose, tests, triglycerides, phospholipids and fatty acid chains. Starch is a biodegradable natural carbohydrate that acts as an energy store in plants and serves as a reserve food supply. Starch is a polysaccharide composed of alpha glucose units linked together by 1 to 4 glycosidic bonds. It constitutes two polysaccharide components, water-soluble amylose and water-insoluble amylopectin. Amylose is a linear molecule and is a long unbranched chain linked together by 1 to 4 glycosidic linkages. Amylopectin is a highly branched polymer linked together by alpha 1 to 4 linkages everywhere and by a 1 to 6 glycosidic bond at the branching points. Starch has a branch structure. This allows for rapid hydrolysis, enabling swift release of glucose for respiration, thereby providing energy quickly when needed. It has a large size, therefore starch remains within the cell, ensuring a localized energy source. It's also insoluble, therefore the water potential isn't affected and prevents any osmotic effects. Cellulose is a biodegradable natural polymer that provides structural support in plant cell walls. Cellulose is a long-chain polymer of glucose molecules joined together. Each glucose molecule is flipped 180 degrees during joining, compared to its adjacent glucose molecules in the chain. This alternating orientation of glucose units results in a straight linear chain. Cellulose is insoluble, therefore the water potential of the cell isn't affected and it prevents any osmotic effects. It also has microfibrils. These are where many hydrogen bonds link parallel strands to form microfibrils, strong fibers. To test for reducing sugars, start with adding Benedict's reagent to the sample, then heat it in a boiling water bath. A positive result will result in a green, yellow, orange or red precipitate, therefore reducing sugar is present. The Benedict test for reducing sugars tests for all monosaccharides and some disaccharides. The non-reducing test tests for no monosaccharides and some disaccharides. The test for non-reducing sugars is as follows. Add a few drops of dilute hydrochloric acid, heat in a boiling water bath, neutralize with sodium bicarbonate, add Benedict's reagent and heat again. If a non-reducing sugar is present, a green, yellow, orange or red precipitate will be formed. To test for starch, add iodine dissolved in potassium iodide to solution and shake or stir. A blue-black color will indicate the presence of starch. To test for lipids, add ethanol and shake. This will dissolve the lipid. Then add water. A positive result will show up as a milky or cloud white emulsion, indicating the presence of lipids. To test for proteins, add burette solution to sodium hydroxide and copper sulfate. If a color change is observed from blue to purple, then protein is present. If there's no color change, then no protein is present. To determine a glucose concentration, these are the following steps. Prepare glucose solutions with known concentrations. This will serve as your standard for comparison. Then perform Benedict tests on each glucose solution. Heat the solution with an excess of Benedict's reagent. Ensure that the same amount of solution is used for each test. After heating, filter out the precipitate that forms. Then measure the absorbance of each sample by using a colorimeter. To do this, calibrate the colorimeter using unreacted Benedict's reagent. Use a red filter for the measurements. Finally, you can plot your calibration curve. On your x-axis is glucose concentration, with your y-axis being graph of absorbance. To test your unknown sample, measure its absorbance, then draw a horizontal line from the value to the point where it hits the curve. Reading down will be your glucose concentration. Guys, apologies for the poor writing on this slide, but I meant to say a triglyceride is formed by the condensation of one molecule of glycerol and 3 fatty acids, forming an ester bond. In triglycerides, there's a high ratio of carbon to hydrogen bonds compared to carbon atoms, so therefore more energy is released than the same mass of carbohydrates. It is also insoluble in water, therefore there's no effect of water potential on the cell. The structure for phospholipid is the same as a triglyceride, however it contains one less fatty acid, and that is substituted by a phosphate-containing group. Phospholipids form a bilayer in the cell membrane. This allows for diffusion of non-polar or small molecules. Phosphate heads are hydrophilic. This means as they are attracted to water, they position themselves towards the aqueous environment on either side of the membrane. Their fatty acid tails are hydrophobic. This means they are repelled by water, and as they do so, they position themselves towards the interior of the membrane, therefore repelling polar and charged molecules. The difference between a saturated and unsaturated fatty acid chain is that in the former there is no carbon-to-carbon double bonds in the hydrocarbon chain, whereas in unsaturated chains there is at least one or more carbon-to-carbon double bonds in the hydrocarbon chain. This is an exam question you might get on the structure of an unsaturated and saturated fatty acid. Make sure to mention all these 3 points to get full marks. Well done for sitting through this video, and now you can have a go at these questions.